Howdy, today I'm talking about binary fission and mitosis. The science understanding we're looking at is, in order to reproduce, cells need to copy their genetic material, then divide to form two new daughter cells. Describe binary fission, describe mitosis, and compare those two. So binary fission is the process that prokaryotic organisms and some very simple eukaryotic organisms use to reproduce. It happens in a few steps. We're going to treat this as if it's a prokaryotic organism, so there's only one chromosome to deal with. So the DNA replicates, which means it copies itself, so you get two copies of the DNA. The cell stretches and gets longer, and then the cell kind of grows a new wall and membrane in between it, and the cells separate. The two cells produced are called daughter cells. This is a very simple process. It doesn't take very long. A bacterial cell can reproduce every half an hour or so, sometimes longer, sometimes shorter. So the DNA replicates, the cell elongates, the cell pinches off, and then the two cells separate. Now each of these daughter cells produced are genetically identical to the cell that we started with, which is important when we're just reproducing cells. So here's an animation showing binary fission. So the DNA replicates, so we get two copies of the DNA. The strands of DNA separate, one going to each cell. The cell elongates, we get the new cell wall and cell membrane forming in between it. The cell pinches off and you end up with two daughter cells. Down here we have, this is a eukaryotic organism that's undergoing binary fission. That looks like a euglena or something like that. And over here we have a bacterial cell doing a similar thing. So DNA replicates, the cell elongates, the cell pinches off, two daughter cells formed. Mitosis is the much more complicated process that most eukaryotic organisms use to reproduce. You do this. There's 4 steps involved: prophase, metaphase, anaphase, and telophase. And then there's a step at the start called interphase, which is just when a cell is sitting around doing what it's doing. So before it starts dividing, a cell is in interphase. So we're going to go through the stages and talk about what happens in the stages of mitosis. So the first thing that happens is in interphase, so when the cell's just sitting around, the first step is that the DNA needs to replicate again. So again, you get 2 copies of the DNA. The chromosome, when it's replicating, is in a stretched out structure called the chromatin structure, which is long and invisible because it's too far apart. When the chromosomes reproduce, they form 2 sister chromatids, and I've got a picture down here. So here we have a chromosome, and once the DNA is replicated, we get 2 sister chromatids joined together at the middle. This is called a centromere here. So the next step is prophase. So in prophase, the chromosomes, they do that condensing down. So normally in interphase, they're long and skinny and stretched out a lot. In prophase, everything condenses down, and you get what's the typical chromosome structure. So if you think of chromosomes, you often think of this kind of an X structure. The other thing that happens is the nuclear membrane breaks down, so the chromosomes are just floating around in the cytoplasm. In the next stage, metaphase, the chromosomes line up along the middle of the cell. This is called the metaphase plate. It's not a physical structure, it's just essentially a halfway point between the cell. We get this mitotic spindle, which is made up of bits of the cytoskeleton. That attaches to the centromere of the sister chromatids. So at prophase, chromosome condenses down, nuclear membrane breaks down. Metaphase, the chromosomes line up along the metaphase plate, halfway along the cell, and the mitotic spindle attaches to the chromosomes. Next stage is anaphase. In anaphase, the mitotic spindle contracts, and that pulls the 2 sets of chromosomes apart from each other. So sister chromatids are separated. So remember, each sister chromatid is genetically identical to the other one. So one half, which contains a full copy of the DNA, goes one way, the other half goes the other way. So we end up with half a cell with all the required DNA for the organism in each half cell, so in each daughter cell. The last stage is telophase, and this is where the nuclear membrane reforms, the daughter cells start pulling apart from each other, and finally you get cytokinesis, which is where the 2 cells break. So cyto means cell, kinesis means break or move. So the 2 cells separate. So anaphase, the sister chromatids are pulled apart to each end of the cell, and then in telophase, the cell elongates, a new cell membrane, new nuclear membrane forms, and then you get cytokinesis, 2 cells separating. So here we have an animation showing mitosis. So we've got the chromosomes condensing, the nuclear membrane breaks down, The chromosomes line up along the metaphase plate here. Then we have anaphase, the sister chromatids are pulled to opposite ends of the cell. Then we have the nuclear membrane forming in telophase, the cell membrane reforms, and then the cell separated cytokinesis. So you need to be able to compare binary fission and mitosis. So in both processes, you start with one cell and you form two cells. And in both processes, the cells that are formed are genetically identical to the cell that you start with. Binary fission is a much simpler process. There's fewer cell structures that need to break down and reform, so we don't have to worry about a nucleus forming, and we don't need to worry about spindle fibers forming. And mitosis, there's a lot more steps and there's a lot more cell structures involved. Like I said, the mitotic spindle and the nucleus. Binary fission and mitosis, depending on the organism. So very simple eukaryotes use binary fission, but all prokaryotes use binary fission. You get two identical daughter cells, like I said. For binary fission, though, mainly that's reproduction of the entire organism. So the whole organism is made out of one cell, so we just use binary fission. With mitosis, this can be used to repair, say, your skin cells. So you get a cut, you need to produce more cells to bridge the gap in your cut. So those cells are produced by mitosis. So it's used for growth and repair as well. It's not just replicating the entire organism. So most of the cells in your body are produced by mitosis. There's another process called meiosis that we'll talk about later. So today on Flipping Science, we looked at binary fission and mitosis. That's it for Flipping Science today. See ya.